Hello, in this video we will discuss about the Fas receptor and apoptosis and what is the Fas ligand. So that is we will also understand about the membrane stress pathway and the extracellular domain as well as related to the mitogen, and there are intrinsic and extrinsic pathway. So the fast induced apoptosis is the extrinsic factors. Intrinsic factor is the another. While the intrinsic factors induced apoptosis and extrinsic factor is also which that is used for the cell death. Cell can be a damaged, injured cell or programmed cell death due to the old cell or damaged cell due to the internal changes. And here is the necrotic and programmed. The necrotic is the injured cell, and programmed cell death is due to the age wise, and the older cell will be die. For example, RBC. So here is the autocrinally due to the internal factor will produce the necrotic factor kappa B and the interleukin, which that is used for the cell death by a cascade of phosphorylation. So this is the necrotic factors. Because the necrotic apoptosis is due to the injured cell and that produce the necrotic factor kappa B, while also it is related to the programmed cell death, because the intrinsic factor will lead to programmed cell death due to the damaged mitochondria. So the mitochondria will not longer to replicate. So here is the another thing is the intrinsic factor. That was the extrinsic factor because the paracrinally and autocrinally which that will be bind. And here is the intrinsic factor means the activation of the BCL2 protein will inhibit the growth factor binding receptor, while the trigger the necrotic factor kappa B as well as intrinsic factor for the mitochondrial damage to produce cytochrome, and use for the intrinsic factor can be mitochondria dysfunction, DNA damage, and older cell. So the significance of apoptosis is that through Fas ligands, damaged or infected cell will die due to necrotic factor kappa B itself binding with cell Fas receptor. Anyhow, the infected cell will be triggered and produce the necrosis factor kappa B will bind with the Fas ligand. After the binding with the Fas ligand, it can be autocrinally or autocrinally or paracrinally, which that is the neighbor cell is also die. So in this way, here is the necrotic factor kappa B is the best example when bind with the NFKB receptor. After the binding, the growth hormone receptor can be inhibit this function. But in this way, the infected cell will be paracrinally bind, and it is the prevention from cancerous cell. If this function will not work, so that means the cancerous cell will be progressed due to the damaged DNA, and there are a lot of things. So here is the plasma membrane and inside is the nucleus. In this way, here is the mitochondria. This mitochondria is, for example, damaged, and the functional mitochondria. So here is the receptor. This receptor contain a monomeric but become a dimeric when bound with a Fas ligand. That is the family of death ligand, the trail and Fas ligand, which that is the death ligand bind, and inside is the death domain, while the extracellular domain is the binding domain. Will trigger the intracellular domain, death domain, to recruit the phosphorylation to activate the Fas associated death domain. And this Fas associated death domain will be bind with the intracellular domain, death domain. So in this way, the disc is also bind the death associated signaling complex. This bind together to give a signal to activate the procaspase 8. This procaspase 8 activation to produce the activated caspase 8 after the cleavage of the proteases enzyme, and there are a lot of things. So after this activated, the caspase 8 will trigger more activation and the cascade of the cleavage formation to the caspase cascade will activate to caspase 3 and this caspase 3 will activate the caspase 9. So the caspase 9 activation will lead to activation of caspase 6. So here is the activate caspase 8 will trigger and with the BCL2, which that BCL2 bond with the bags. So the BCL2 is the gene activation to produce cytochrome C efflux due to the bags binding with the mitochondria, damaged mitochondria, and cytochrome C will be efflux. So here is the caspase 9 will be activate through caspase 3 activation, and this caspase 9 activation will trigger the caspase 7 activation, and 7 activation will lead to trigger the 6 and 2. 6 and 2 activation will lead to caspase 6 will activate the caspase 8 and 10 again, that is 8 produce, and in this way the 8 will again use same mechanism. And here is the APAF protein which that is bind, and this is a multimeric protein will bind with the cytochrome to produce the apoptosome. This apoptosome is used for the apoptosis, but we will not understand about that things the detail with the detail. So here is the DNA. Let's begin to understand the intrinsic pathway in the nucleus. So for example, the reactive oxygen species will produce and damage the DNA. After the damaging of the DNA, activation of the atom phosphorylation and the transcription factor will uh, trigger the CHK2, and the CHK2 will bind and the MDM2 and P53 will be triggered to replicate transcribe, and this P53 will be produced and the NOXA, BAX and PUMA will be produced. These three type of protein will trigger the P53 will bind with the BCLX1, and in this way the BCLX1 will be separate and the P53 will be separate, which that will be trigger the bags binding with the BCL2 will be separate, will be not separate but trigger an activation, and in this way that will be bind with the mitochondria to efflux of the cytochrome and again the APF1, the apoptosome formation. So let's begin to understand about the membrane stress pathway. So the membrane stress pathway is the ceramide production from the membrane. Ceramide production due to the different damage or toxins. So in this way the production of these will trigger the kinase activation for the proliferation inhibition and mitosis inhibition as well as the maturation inhibition. So here is the mitogen which that is responsible for the proliferation, cell cycle, mitosis and meiosis. But the kinase activation of the kinase protein due to the ceramide production will inhibit, while the cyclin dependent kinase and cyclin will be inhibit. So the mitosis will be inhibit and effector 2 factor protein and retinoblastoma protein will be not dissociate. So that means the mTOR C1 and 2 will be also inhibit and mitogen will inhibit to apoptosis. Thanks.